But of course, the world in which I have been growing up as a young scientist has been a world without borders. The topic that we are working on, which is biomedical research, is just too broad to allow progress in one single country to be relevant. And so we need this international collaboration for the sake of humanity. So science should have no borders. And it is, in a way, very sad that there's this tendency to split the world into blocks of influence that make it more difficult for scientists to circulate and to communicate among each other. Well, of course, when we think about death, we immediately think about negative feelings because our existence will finish for each of us with death. And so it looks like a fearful moment. But when you study medicine, you learn very early that actually our cells are prone to die individually, and that cell death is actually occurring during embryonic development as part of the plan to shape our body and our organs during embryonic and fetal development. And that even in the adult organism, to maintain tissue homeostasis, cells have to be turned over, and that cells have to die to allow for the renewal by proliferation of fresh cells. And so cell death is absolutely necessary for homeostasis of the adult individual. And as such is something that is natural, that has to be studied with positive feelings. And beyond that, of course, cell death can occur in an excessive way or with the wrong program, with consequences that are not physiological, and then play a role in multiple different diseases. Cancer, which is affecting 1/3 of us during our lifetime, can be interpreted as a disease in which cell death that should have occurred and that should have eliminated a premalignant cell has been somehow suppressed. And then the forgotten cell death or the suppressed cell death is pathogenic. So it's the deficiency of the lethal pathways that leads to malignant disease to some extent. And then there are many diseases in which excessive cell death occurs. So when we talk about, let us say, neurodegeneration, there are too many neurons that are suppressed by cell death pathways that have not been planned by evolution. And this excessive cell death will then lead to a reduction of organ function, in this case, the central nervous system. And in the same way, you can have slow degenerative processes in multiple different organs, in the heart, the kidney, liver, or you can even have sudden instances of cell death due to external influences like intoxication or absence of oxygen or absence of vascularization and blood flow that will be pathogenic more suddenly. So there are chronic and acute diseases that are due to excessive cell death. Yeah, so we can say roughly that in the context of excessive cell death, of course, the therapeutic goal is to make cell death less probable to occur and to protect cells against unwarranted cell death. And in the case of oncology that you mentioned, the goal of therapeutic interventions frequently is to induce cell death in malignant cells and hence to eliminate them. Now, it is not that simple. It's not just about apoptosis, which is one of the modalities of cell death. It's about eliminating the cells that contribute to the formation of a tumor, which are multiple different cells, but in this case, of course, we are interested in malignant cells, so the cancer cells, in a way that they don't just disappear silently, but that they actually are able to stimulate an anti-cancer immune response. So the purpose of a therapeutic intervention with so-called cytotoxic agents, which can be chemotherapeutic agents, or that can be targeted therapies, or it can be also irradiation in radiotherapy. The purpose of these cell death-inducing therapies at the end is to induce an anti-cancer immune response. And if this does not occur, the malignant cells can be replaced very rapidly by proliferation, and the therapeutic intervention is, in a way, inefficient. So the purpose, of course, is to treat the patient and then to discontinue the treatment, because all treatments have side effects. They cannot be administered forever. So if we want that the cancer treatment has long-term effects beyond treatment discontinuation, the immune system of the patient has to kick in and has to take care of the so-called residual cancer cells, those that have not been killed, to keep them in check and to prevent the tumors to strive, to relapse, to metastasize, and ultimately to seal the fate of the patient. So let us start with the definition of the hallmark. So a hallmark of aging is a biological characteristic of the system that accompanies aging, logically. So it has to be more accentuated when an individual ages. And then there's also a functional implication. So if we experimentally enhance a hallmark of aging, we should be able, in a model system, to accelerate the aging process, and more difficultly, if we experimentally suppress or attenuate a hallmark, in that case, we should expect that the aging process is decelerated or even perhaps halted in its evolution. So using these characteristics or this definition of the hallmarks of aging, we came up with a list of 14 intertwined processes that accompany the aging process that can be experimentally manipulated to enhance or reduce the pace of aging. And so we don't have time to enumerate all the 14 hallmarks, but basically they affect any component of the cells in our body. So when we talk about DNA or RNA or protein or organelles, they are all affected during the aging process. They affect the supracellular functions, so how cells communicate in the organism, and they even affect our psychosocial integration in the society. So aging is a very complex process, and I chose, that's my particular selection in this among these hallmarks, to concentrate our efforts on one particular hallmark of aging, which is disabled autophagy. Autophagy is a process in which portions of a cell are sequestered in membranes within the cell. They are called autophagosomes, and then destroyed by lysosomes. So it's a little bit imaging autophagy is a Greek neologism that refers to self-eating. And so if the cell was a body, a human body, by comparison, then when it eats portion of itself, the mouth is closing on the sequestered portion of the cell, and then it will fuse with the stomach of the cell, which is a lysosome, to allow for the digestion of the luminal content. And so this process, which sounds very awkward because self-eating sounds a little bit strange, isn't it? Is extremely important to turn over portions of the cell. So I talked before when I introduced the topic on cell death on the importance of cellular turnover for the tissue homeostasis in the adult organism. And within each of our cells, there is a turnover of aging and dysfunctional organelles and proteins, protein complexes, and other macromolecules that is accomplished by this autophagic process. So portions of the cells self-destroy to allow for renewal. In a way, you can visualize the autophagic process as a mechanism of an anti-aging mechanism that allows for the self-renewal and turnover of the aged proportions of the cell. Fortunately for us, the control of autophagy is accomplished by several internal and external factors. So usually as we age, the capacity of the autophagic process is declining, and we can reactivate autophagy by pharmacological tricks, by inactivating what we call autophagic checkpoints, so the endogenous inhibitors of autophagy, some of which are intracellular. So for instance, the mTORC1 complex, which is an intracellular protein complex that can be inhibited by rapamycin and similar molecules such as the so-called rapalogs. So these are small inhibitors, pharmacological agents that act on the mTOR complex to inhibit it, and the inhibition of a complex that suppresses autophagy then allows for the stimulation of the process that we're interested in. And so there we also have extracellular checkpoints of the autophagic process. So one example is insulin and insulin growth factor. Another one is a protein that we were working on that is called ACBP, for acyl-CoA binding protein. And it also has other names. And if we inhibit insulin-like growth factor, if we inhibit ACBP, which are extracellular tissue hormones, we can reactivate deficient autophagy and hence revert the age associated decline in the autophagic process and stimulate an anti-aging pathway. Well, I think in this case it is there's no a clear medical definition of a disease. It is more a practical definition. So the Federal Drug and the Food and Drug Administration, FDA, in the US, and the European agency, the EMA, they allowed to intervene on diseases therapeutically. So if you want to perform a clinical trial, if you the objective of your research is to obtain a drug that can be commercialized and at the end will receive the approval by the FDA or the EMA, you must treat a disease. And so until a few years ago, for instance, obesity was not considered as a disease, and hence obesity could not be treated as such. The only thing that you could treat was the obesity-associated diseases like diabetes, for instance. So this has changed, and since obesity now is considered as a nosological entity or as a disease, the FDA and the EMA have allowed to perform clinical trials just with the purpose of reducing the weight of individuals, reducing the body mass index. And this, of course, has changed radically the way how large portions of the population in the USA or in Europe are now treated for obesity. And in the same way, aging is not considered a disease at this point, but it might be. And if the FDA or the EMA defined what they consider among the external manifestations of aging as a biomarker of disease. So let us say sarcopenia, not as a pathological end-stage manifestation, but a reduction of muscle strength or reduction of what is called the cognitive abilities before you actually manifest Alzheimer's disease. So the functional capacity or intrinsic capacity of an individual that declines with aging, even before it reaches a level of deterioration that is classically defined as a disease, then it would be able to administer drugs or treatments that are not drugs. That could be exercise or specific diets in a way that are FDA approved because they prevent the functional decline that is linked to the aging process. And of course, the community of researchers that are interested in aging, the so-called geroscientists, and the specialists in what we can call geromedicine, which are not geriatricians, that are people who are interested in treating aging as a condition that requires a therapeutic intervention. These people try to influence the FDA and the EMA in the sense that the functional decline accompanying aging would be considered as a condition that has to be treated. Maybe you could call it a disease, but this is more about the legal terms of the FDA and EMA than anything else. Well, actually, caloric restriction is probably the only intervention that across all known organisms in nature is prolonging health span and lifespan. So if you subject yeast cells or worms or birds or plants to caloric restriction, you tend to increase their health span and lifespan, talking about a reduction in calories that does not induce any harm. So it's about thresholds also. Too much caloric restriction is, of course, inducing cachexia at the end, and it kills by hunger. So we're talking here about thresholds, and you have to imagine that initially it was thought that life is like a chemical reaction. So if you slow down the chemical reaction by reducing the temperature, or by reducing the amount of substrate that can be catalyzed in this reaction to generate an end product, then you would increase lifespan. And this theory actually turned out to be wrong. It is not just about the velocity of reaction. It's also about the way how the organism responds to a type of stress that can be useful, or that can be positive. This is also called eustress, and which is this small reduction in caloric intake that, for instance, stimulates autophagy. So if you take a mouse and give no calories for a night, then mice usually are active during the night. So if you would deplete food from a mouse during an entire night, that mouse will lose 10% of its weight. And this induces autophagy in practically all cells of the organism, with the exception of the central nervous system that is buffered against this caloric restriction-induced autophagy. In a human being, you have to do it for a longer period, because our organism is more resistant against short-term caloric restriction or food depletion. But at the same, after a week or so, you also get autophagy in most cells in the body that you can study, for instance, in skin biopsies, or more easily in circulating blood cells. So the effect of caloric restriction is stimulation of stress responses, one of which is autophagy, that at the end slow down the aging process. So the idea is that since it is very difficult to achieve that an individual reduces his or her caloric intake in a voluntary fashion, it would be possible to give natural agents or drugs that achieve a similar effect than caloric restriction without the need of reducing caloric intake, and to change your lifestyle. So there are some agents that achieve this process that we call caloric restriction mimetics, that are biochemically very different. So it's not a class of agent that you could define by specific molecular properties or biophysical similarities. It's about their capacity to induce autophagy in a non-toxic fashion without harming your cells, and hence stimulating the anti-aging pathways that are usually only achieved if you reduce caloric intake. And so, for instance, one of them is quite interesting. It's spermidine. Spermidine is a natural polyamine that is actually contained in all our food items. Also, the amount of spermidine is quite heterogeneous. So, of course, you have no spermidine is usually associated with DNA. And so if you take in DNA-free food like pure sugar or milk, there's very little spermidine contained in these food items. But if you eat fish or vegetables which are full of the nuclei of the cells that you ingest, there is much more spermidine contained in these food items. And so we could show in mouse experimentation that external supplementation of spermidine can actually stimulate autophagy and then increase health span and lifespan. This is something that is evolutionarily conserved, so we can give spermidine to yeast and to C. elegans and to Drosophila, so little worms or flies, and obtain the same effect. And presumably, spermidine can also have positive effects on human health, although no serious clinical trials have been done yet in this. There's mostly about retrospective analysis of the amount of spermidine that you consume with your food that are associated with a positive outcome with respect to cardiovascular health or the reduction of the propensity to develop cancers, or even the reduction of neurodegeneration. Now, spermidine is also, that's something that we discovered very recently, together with Frank Madeo, who is my collaborator in this area of research. It turns out that actually, if a mouse or a fly or a worm is restricted in caloric terms, the cells in this organism produce more spermidine because it's an endogenous metabolite through pathways that we have to understand. And this surge in spermidine that occurs in an endogenous fashion is then stimulating autophagy. It must occur so that autophagy can be induced and to mediate the beneficial effects of caloric restriction. So in a way, spermidine is an endogenous metabolite that acts as a second messenger to confer the benefits of caloric restriction. And then we have other agents that can stimulate autophagy that are less natural. So for instance, so-called chalcones that also occur in nature, but not as endogenous metabolites in our organism, that can be given like drugs and that stimulate autophagy to increase health span and lifespan in model organisms, including in mice. Actually, the word precision medicine has been mostly invented in the context of oncology. So you know that each cancer is different because the genetic makeup of the cancer cells that develop spontaneously in an individual is heterogeneous and may require specific interventions that depend on the mutational status of the cancer cells. So you will not treat a cancer that is due to the mutation of a growth factor receptor like EGF receptor in the same fashion as you treat a cancer that is due to the mutation of a DNA repair enzyme. And so for that reason, there are specific drugs that have to be administered as a function of the diagnostic exploration of the genetics of the cancer cells. So this is called precision medicine or precision oncology. And in the same way, we can talk about precision geromedicine because in a genetically heterogeneous population like the human population, so we are not talking about laboratory mice that are inbred, we are talking about the outbred heterogeneous human population. There are genetic predispositions and environmental factors that are specific for each individual and that then modulate the aging process, giving rise to age-related diseases. Most diseases are time-dependent in their manifestation, so the vast majority of diseases are actually age-related. But we all develop, to a certain extent, different age-related diseases, and this requires specific interventions. I'm not keen to treat the age-related diseases. I think we have to treat the underlying causes of these diseases, but still the causes are to some extent heterogeneous and hence may require individual specific interventions. And so if we talk about precision geromedicine, this is only possible if we have a diagnostic arsenal, like in oncology, that allows to detect these specific pathways and genes and individual exposures to environmental factors that then are linked to a specific intervention. So we're talking about precision diagnosis, and that leads then to a specific or precise therapeutic intervention. So it's only possible if we link up multi-omics. So that's the idea that we measure many different parameters in an individual beyond that, what is usually done by a general practitioner, and then intervene on the age-related and age-inducing pathways that are specific for an individual as far as we can by a new tailor-made arsenal of therapeutic agents. So of course, we have to distinguish two different ways to measure the span of life. So it's health span versus lifespan. So health span is the time in your life that you spend without major handicaps in a healthy fashion. And lifespan is just the measurement of the duration of your life between your birthday and your last day. And so we know, of course, that the health span is the goal that we want to achieve. We want to have a population that lives a longer and healthier life without populating the geriatric departments in our hospitals. So the problem that may occur if we extend lifespan, that's a theoretical problem. It's just a sort of theoretical consideration. So the problem that may occur is that if we extend lifespan without extending health span, that the period of invalidity and unhealthy suffering of individuals will be extended. And that's, of course, something that we don't want. We want to increase the health span. Well, that's actually the goal of geromedicine. It's not about getting 100 years old. The goal is to get to 99 years and a half in a good shape and to have a very short period of terminal illness. And this is, of course, also a theoretical goal that has to be achieved by a judicious administration of medical interventions. Already in the Oath of Hippocrates, it is foreseen that the doctor that treats a patient should aim at maintaining vitality without exaggerating the therapeutic measures that lead to an extension of life that is associated with suffering. So I'm not advocating here for euthanasia. That's not the purpose here. It's about what we do, to which extent, with which intensity, in a context of precision medicine, to a maximal extension of the intrinsic capacity of an individual. As you can talk about also fitness, so physical and mental, cognitive fitness that allows to live a meaningful and happy life. And that's the purpose of geromedicine. So it's not about geriatric interventions in unhealthy individuals. But I think it's about division of labor. So the more sophisticated a society is, the more specialized are these professions, and the more divided are the tasks, the division of labor. So as a scientist, of course, I should concentrate on the problems that I want to solve, or even more importantly, the problems that I have to invent. So the questions that I have to ask to Mother Nature, first have to be invented, and then have to be answered. And that's actually the purpose of my profession, to explore the space that is infinite, with interesting questions that can be addressed by scientific methods, and then lead to new insights that may be useful for biomedical applications. And so I consider that this is the central part of my job. Communication should be on top of it, but not a single individual can do every task that are related remotely to his or her purpose in life. And so I think it is very good that there are trained persons who are able to explain to the general public what is science about, what is medicine about, what are the recent areas of progress in the biomedical space, and to which extent that may be useful for society. So that's my point of view.